There is one more 8 marks question left in this chapter. Let us start with this topic and the 8 marks question. The name of the 8 marks question, very important for examination point of view. This year we can expect this question. Life cycle of Entamoeba histolytica. Entamoeba histolytica is basically, if you see, it is a microscopic parasite and a monogenetic parasite. What do you mean by monogenetic parasite? It completes its life cycle only in one host. It inhabits to live inside the large intestine of a man and causes a disease called amoebic dysentery. What is that called? Amoebic dysentery or amoebiasis. Mode of infection, how it causes? It causes or spread through contaminated food and water. Basically, houseflies, cockroaches act as a mechanical vector for this disease. It is cosmopolitan in distribution, but more common in tropical regions. More common in tropical and subtropical regions of the world. It is very common in the rural areas and urban areas where there is a huge population. So, let us see the structure of this parasite. It passes through 3 distinct stages in its life cycle. First one is tropozoid stage. The first life cycle, tropozoid stage. Second one precystic. Third one cystic stage. How tropozoid stage looks like? It is general characters of this tropozoid stage. Later on we will see the structure and its shape. It is the most active and feeding stage of Entamoeba histolytica and very pathogenic stage of his life cycle. At the same time, it lives in the mucosa and submucosa layers or membranes of the large intestine in man. It is moved with the help of single blunt finger-like pseudopodium that is called as a lobopodium. What is that called? Lobopodium. So, this pseudopodium helps to move the animal from one place to another place. The body is covered by thin plasma lemma. Cytoplasm is differentiated into outer clear viscous non-granular ectoplasm and the inner fluid like granular endoplasm. So, cytoplasm was differentiated into these many types. Now, let us see here. See these, what are the cell organelles that are present in it? Ribosomes, food vacuoles, and one nucleus, which is cartwheel shaped nucleus, is present in the endoplasm. However, contractile vacuoles, endoplasmic reticulum, Golgi apparatus, mitochondria were absent. These cell organelles are absent. Due to absence of mitochondria, it shows obligatory anaerobic character. You can see the structure of this tropozoid, pseudopodium, okay, endoplasm, ribosomes, food vacuoles, and here you can see the cartwheel shaped nucleus and vacuoles with RBC, right? See, the absence of mitochondria, already said you, it shows obligatory anaerobic nature. It produces an enzyme called histolysin, due to which its species name is called as histolytica. What is that called? Histolytica. Now, due to the effect of this enzyme, mucosa, submucosa layers, mucosa and submucosa layers of large intestine dissolved and releases the blood and tissues and tissue debris which are ingested by the tropozoids. Now, hence the food vacuoles, whatever present in this tropozoid, contain erythrocytes or red blood cells. Okay. Apart from that, some of the fragments are also present tissue fragments, etc. Fragments are also present, and epithelial cells are also present. Now, mode of nutrition. Holozoic nutrition is present, right? Presence of RBC and in food vacuoles and cartwheel shaped nucleus are the chief and important characters of trophozoite. Let us see the pre-cystic stage, non-feeding stage, non-pathogenic stage of the Entamoeba. It commonly a non-motile and non-feeding stage. Okay, it lives in the lumen of large intestine. Okay, now it is a small non-motile spherical, at the same time oval shaped, okay, non-motile structure. The cytoplasm of this structure stores, diagram I will show you in the next slide, glycogen granules. What it shows? Glycogen granules. So these glycogen granules, okay, these glycogen granules and chromatid bars, what are they? Chromatid bars made up of ribonucleic proteins. These ribonucleic proteins act as a reservoir for food. Apart from this, if you see the structure, this is the pre-cystic stage structure. Now, cystic stage, how it looks like. It is round in shape, at the same time surrounded by thin, delicate and highly resistant cyst wall. What is that? Cyst wall. Cyst wall is present. Okay. Now it is found in the lumen of large intestine. The process of development of cyst around this structure is nothing but encystation, which means helps to this encystation process helps to this parasite to overcome unfavorable conditions while it is entering into the new host. Now the nucleus undergoes successive two mitotic divisions. Okay, if mitotic division happens, what happens? Finally, two new individuals will be produced at the end of the mitotic cell division. Okay, two mitotic cell divisions leads to formation of four daughter nuclei. How many nuclei? Four daughter nuclei. This type of cystic stage is called tetra, means four, nucleate cyst. Finally, let us see the structure of cystic stage. You can see the cyst wall. This is a cyst wall. Okay, looks like a cell wall or cyst wall. Okay, cyst wall, chromatid bars, glycogen granules are present in it. Life cycle. Trophozoite undergoes two binary fissions. It undergoes two. You know very well about binary fission. Bi means two. Okay, trophozoites divides into two equal sized structures in the wall of large intestine. Produces finally daughter Entamoeba. Okay. Finally, they feed upon the bacteria and host tissue elements. Now, after repeated binary fissions, trophozoites are normally increases in size. Okay, some of the young ones enters into the lumen of large intestine and transforms into pre-cystic stage. Okay, after formation of pre-cystic stage, it will transform into a cystic stage and produces tetranucleate cyst. What it produces? Tetranucleate cyst. This entire process completed only within a few hours. After that, tetranucleate cyst comes out along with the excretory products and live for 10 days. How many days? 10 days. After that, the cyst wall reaches new host. The cyst reach the new host through contaminated food and water. They passes into a new human host. Okay, if they reach to new host, okay, where the cyst wall gets ruptured, the cyst wall whatever it is present, this cyst wall gets ruptured. After rupturing, the action of trypsin enzyme takes place. This trypsin enzyme leads to releasing the tetranucleate amoeba. Now, such existed tetranucleate amoeba is called metacyst. What is that called? Metacyst. Four nuclei, what are they? Four nuclei of the metacyst. Four nuclei of the metacyst undergoes to mitotic divisions and produces 8 pronuclei. But it produces 8 pronuclei. Each nucleus gets a bit of cytoplasm. Each nucleus gets a bit of cytoplasm. Okay. Thus, 8 daughter Entamoeba. Okay. Daughter Entamoeba or metacystic Entamoeba tropozoites are produced. Okay. Now, finally, 8 are produced. After this, these young ones develops into feeding tropozoites. Again, the life cycle will be repeated. They invade the membrane of mucosa and submucosa and develops into mature tropozoites. Now, the tropozoid dissolves the mucosal lining with the help of histolysine. Again, the cycle repeats. Go into the deeper layers of the mucosa and submucosa and causes ulcers. Now, ulcers contain various types of cellular debris like lymphocytes. What are they? Lymphocytes, red blood cells, bacteria, etc. Finally, it leads to formation of abscess. What is that? Abscess. Okay, infection in the wall of the large intestine. Ultimately, it leads to blood with mucus in the stools or excretory products. Now, this condition is called as amoebic dysentery. What is that? Amoebic dysentery. Now, this amoebic dysentery or amoebic dysentery, also known as intestinal amoebiasis, or tropical amoebiasis. Some people do not exhibit symptoms. Such people are called as basically carriers or asymptomatic cyst carriers. Finally, what is the prophylaxis for this disease? Using boiled and filtered water always, and washing hands and washing fruits and vegetables before going to eat, and also